Dear students, in today's lecture, let's make an attempt to know about role of biofertilizers in mulberry production. Introduction. During the year 2013-14, India produced 26,480 metric tons of mulberry raw silk, and in order to meet the requirement of producing such huge quantity of raw silk, production of sufficient quantity of quality mulberry leaf is a prerequisite. Application of chemical fertilizer for achieving steady mulberry crop production will also demand more attention. Presently, in India, the requirement of nitrogen fertilizer is in the tune of 76,650 metric tons per annum to meet the requirement of approximately 2.19 lakh hectares of mulberry at the rate of 350 kg nitrogen per hectare per year application rate. This huge requirement is also due to the introduction of high yielding mulberry varieties like S36 and B1. Similarly, the requirement of phosphorus is approximately 30,500 metric tons per annum. This vast requirement of nitrogen and phosphatic fertilizer cannot be fulfilled merely by depending on the application of synthetic fertilizers because of their scarcity, escalating cost and high demand for the production of food crops by the agriculture sector. Since mulberry leaf production is largely dependent on the application of chemical fertilizers, the production cost of leaf per unit area is also increasing day by day. Due to the escalating cost of the fertilizer, especially small and marginal holders are unable to apply them as per the recommendation 300 to 350 kg nitrogen, 120 to 140 kg phosphorus and 120 to 140 kg of potash per hectare per year for K2, S36 and V1 mulberry varieties respectively. As applied partly and frequently lose cocoon crops. Even if the nitrogen and phosphatic fertilizer are applied adequately, the plants cannot absorb the full amount of nitrogen because of volatilization and leaching loss. Similarly, most of the phosphatic fertilizer applied in tropical soils remain unutilized as insoluble phosphorus due to its quick fixation, and only 15 to 25% of the phosphorus is utilized by the plants. As the farmers are not getting the full benefit of the chemical fertilizers, especially nitrogen and phosphorus applied, application of the same through biological means in the form of biofertilizers in the key role to sustain sericulture productivity. Although in any soil microorganisms are naturally available, their population is very much scanty in most of the soils, which may not be sufficient for agriculture activity. Besides, the role of VA mycorrhizal fungi in the improvement of growth and mineral nutrition of agriculture crops have also attached a lot of attention of the researchers worldwide. So, in order to harness the potential benefits from these organisms, the artificially cultured microorganisms are used either or fixing atmospheric nitrogen or solubilizing insoluble phosphorus or mobilizing available phosphorus and other plant nutrients to the beneficiary crop. They are biofertilizers. In other words, biofertilizers are the nutrient fixing or trapping microorganisms which absorb nitrogen, phosphorus, potash, etc. and make them available to the plants. Some of them are symbionts and some are free living. Use of different biofertilizers in mulberry. Number 1, Azotobacter biofertilizer. It is a bacterial preparation of live Azotobacter cells blended with suited carrier materials like lignite, peat, charcoal in powder form. It is capable of supplying nitrogen to the plants through biological nitrogen fixation and enhanced plant growth. The material is commercially available as SERICULTURE. Azotocrocacum a free-living nitrogen-fixing bacteria isolated from the rhizosphere soil of mulberry is found to be highly suitable for manuring mulberry garden as a supplement to nitrogenous fertilizer under various agroclimatic conditions. In most of the mulberry garden soils, Astrobacter crocacum has been found as a dominant species. In general, they are pleomorphic. The size of young and raw-shaped cells varies from 2 to 7 into 1 to 2.5 microns. The morphology of cells is dependent upon the composition of medium. Astrobacters are known to form cyst, which accumulates poly-beta hydroxy butyric acid and able to give vegetative cells after the onset of favorable condition. It is soil inhabitant, cyst former and moderate slime producer with brown to black pigmentation. The population of Astrobacter in the rhizosphere of crop plants and in cultivated soil is generally low, which are not sufficient for proper atmospheric nitrogen fixation. To overcome this limitation, repeated application of Astrobacter biofertilizer is required to increase the number of biobacteria to 10 to the power of 7 to 10 to the power of 8 per gram soil for maximum nitrogen fixation. Application of Astrobacter biofertilizer in mulberry. Astrobacter biofertilizer at the rate of 20 kg per hectare per year in 5 equal splits, that is 4 kg per hectare per crop mixed with 200 kg of powder FOM is applied in between the rows of mulberry within 10 days after leaf harvest or pruning and intercultural operation by making furrows. The furrows are immediately covered after application of biofertilizer and irrigated. Astrobacter can be used before or after the application of chemical nitrogen fertilizers with a gap of at least 10 to 15 days. For B1 mulberry variety, after or before Astrobacter fertilizer application, NPK should be applied at the rate of 175:140:140 kg per hectare per year in 5 equal splits. Phosphate solubilizing biofertilizer. Phosphorus is the second most important plant nutrient element next only to nitrogen. However, the availability of applied phosphorus is only 15 to 20% due to its fixation is soil into non-available forms. Its availability can be increased by using phosphate solubilizing biofertilizer called SERICULTURE. It contains live cells of Bacillus megatherium. SERICULTURE increases the availability of phosphorus to the plants and increase phosphorus use efficiency. It helps in profuse root development and also increases leaf yield by 10 to 15%. It produces plant growth promoting substances. SERICULTURE can be used along with a mixture of rock phosphate and superphosphate 1:1 or with rock phosphate alone to reduce the cost of fertilizers. SERICULTURE is recommended to be applied at the rate of 5 kg per hectare per year in 2 to 5 equal split doses. The required quantity of SERICULTURE is mixed with 200 kg of well decomposed farmyard manure or compost or vermicompost and applied in furrows between mulberry rows and covered with soil followed by irrigation. For 2 times application, the first dose of SERICULTURE is applied during June to July immediately after pruning and the second dose after 2 leaf harvest. For 5 times application, the required quantity of SERICULTURE, that is 5 kg per hectare per year, is applied in 5 equal split doses after harvesting or pruning of each crop mixed with 200 kg of farmyard manure. For use in nurseries, the basis of prepared mulberry cuttings can be dipped in slurry of SERICULTURE prior to planting. While transplanting the saplings, the roots should be dipped in a slurry of SERICULTURE and planted. For better results, a recommended dose of farmyard manure should be applied in 2 equal split doses. Serifos can also be used along with nitrogen fixing biofertilizer, that is Prakriti, Astrobacter, Azospirillum, etc. for synergistic effect of mulberry. Serifos should not be exposed to direct sunlight or heat. It should be always stored in a cool place. It should be used before the expiry date. It is very important to maintain proper soil moisture for multiplication of the microorganisms. Chemical fertilizers should be applied 2 weeks after the application of Serifos. However, Serifos and rock phosphate can be applied together. It is advised to consult expert before applying pesticides to the soil when Serifos is used. VA mycorrhiza. The recommended dose of phosphorus for mulberry under irrigated conditions is 140 to 120 kg per hectare per year for optimum leaf yield. However, most of the phosphatic fertilizer applied to soil get fixed and are not available for plant growth. Further, small and marginal farmers in India are economically not sound to buy and apply costly phosphatic fertilizer. Under such circumstances, inoculation of mulberry with certain efficient strains of vesicular aspergillar mycorrhizal fungi is highly beneficial. Mycorrhiza literally means fungus root. VA mycorrhiza is a symbiotic association between the plant root and certain useful fungi, which forms vesicles and arbuscules in host root cells. They colonize the root system of higher plants, especially under phosphorus deficient condition, and greatly enhance the absorptive surface area of root system and nourish plant through other micronutrients and phosphorus in particular. There are several types of VA mycorrhiza fungi, of which Glomus mosaic and Glomus fasciculatum are found highly beneficial for mulberry. VAM is available as soil based inoculums containing mycorrhizal spores, hyphae and host root pigments. It is recommended to use VAM in mulberry nursery to raise mycorrhiza inoculated saplings for new plantation. The soil based inoculums of Glomus mosaic or Glomus fasciculatum or mixed culture of both can be used in mulberry beds to raise inoculated saplings. 200 kg of soil based inoculums, that is 20 spores per gram dry soil, is required to raise requisite amount of saplings to plant 1 hectare of land. About 5 to 6 months old inoculated saplings can be transplanted to the main field to raise new plantation along with surface soil of nursery, that is 15 cm depth. 1 inoculated saplings are used for new plantations, no more inoculation is required. After transplanting the nursery raised saplings with VA mycorrhiza in the main field, phosphorus should be used at the rate of 30 to 35 kg per hectare per year instead of 120 to 140 kg per hectare per year. Farmyard manure should be used at the rate of 20 metric tons per hectare per year. Nitrogen and potash fertilizers can be used as per the recommended dose. Irrigation should be followed as per the recommendation. The extent of symbiotic association of VAM depends on the fertility level of soil. High fertility of soil leads to the low mycorrhizal association and poor symbiosis. Many tropical crops and pastures are grown on soil that are very deficient in phosphorus, often slow good root colonization and better yield compared to non-mycorrhizal plants. Of the various microorganisms colonizing the rhizospheres, VAM fungi occupies a unique ecological position as they are partially inside and partly outside the host. The internal phase of the mycorrhizal fungus does not encounter competition and antagonism from other soil microorganisms, but get an ensured source of nutrients from the host. This advantage enables them to achieve a large and more functional biomass in more intimate contact with the root and thus increase their chances of exerting a greater effect on plants than other microbial species restricted only to rhizosphere. Thus, WAM association to the plant not only ensure the increased availability of water and nutrients, it also helps in the uptake of microelements, alterations in the physiological activity of host, increases the level of amino acids in colonized roots of several plant species. These association also plays an important role in biological control of root pathogens, affects the growth of plants by production of hormones and antibodies. VA mycorrhizal fungi also offers the resistance in plants to fight against leaf and shoot pathogens by providing strong vascular to fight against the vascular pathogens. Similarly, the crucial role of the WAM in reducing the nematode population by the production of chemical, physiological and morphological changes in host plants have been well documented. VA mycorrhizal inoculation technique. Two types of WAM inoculation procedure can be followed. Number 1, in the nursery, for raising mulberry saplings to take up a new plantation, and number 2, an established mulberry garden can be inoculated with WAM by intercropping techniques. Precautions. Phosphatic fertilizer should not be used in nursery bed before and after inoculation. Phosphatic fertilizers in reduced dose can be used only after 4 to 5 months of inoculation. Urea can be used at the rate of 1.735 kg per bed only after 2 to 3 months of planting as a booster dose. No pesticide or fungicide should be used in soil during inoculation. These can be used if required at least 1 month before inoculation and should be well drenched with water to rule out the residual effect. Inoculum should be effected from authentic source. WAM is eco-friendly, economical and easy to adopt. It can be reduced 15 to 20% of recommended dose of phosphatic fertilizer without any adverse effect on leaf yield and quality. It saves approximately 563 kg of single superphosphate per hectare per year. Only one inoculation is required. The use of WAM can be reduce the incidence of root pathogens. Precautions in field use of the biofertilizers. Chemical fertilizers, especially nitrogen, should not be mixed or applied along with azotobacter biofertilizer. There should be a gap of at least 10 to 15 days between the application of the biofertilizer and the chemical fertilizers. Biofertilizers should be kept away from heat and direct sunlight and should be used before the expiry date. After application of biofertilizer, the land must be irrigated to save live bacterial cells from desiccation. Biofertilizers should be collected from an authenticated source to avoid spurious materials. Precautions in the field use of WAM. The starter culture of the required WAM fungi for mass multiplication should be collected from an agriculture university or an authorized agency or institute where research on WAM mycorrhiza is going on. No fungicides or pesticides should be used during the preparation of WAM inoculum. No phosphorus fertilizers should be used during the WAM production. Irrigation should be followed in every 5 to 6 days for good growth of host plants. The prepared inoculum should be used within 1 to 2 months, preferably immediately to get best results. While production of WAM inoculums, care should be taken to avoid multiplication of insects or any type of soil-borne pathogens or pests, especially root nematodes. Hence, before preparation of the inoculums, Soil fumigation or sterilization by burning is a must to avoid all these. The worm inoculum should be tested for contaminated with pests or pathogens. It is better to avoid transportation of large quantity of soil based worm inoculums from far off place. Instead, only starter culture free from pests or pathogens should be procured and multiplied in the place where it is to be used. Biofertilizers are the nutrient fixing microorganisms which absorb nitrogen, phosphorus, potash, etc. and make available to the plants. In mulberry biofertilizers such as Azotobacter, phosphate solubilizing biofertilizers and WAM are recommended. Azotobacter fertilizer is applied at the rate of 20 kg per hectare per year in 5 equal splits. Phosphate solubilizing biofertilizers such as SERICULTURE is recommended at the rate of 5 kg per hectare per year in 2 to 5 equal split doses. It is mixed with 200 kg of well decomposed FOM or compost or vermicompost and applied. For WAM, 200 kg of soil based inoculum is used to inoculate cuttings required for raising saplings to plant 1 hectare of land. For inoculation of 1 hectare established mulberry garden, the requirement of WAM inoculum is 1000 kg and is inoculated through raising maize, sorghum, ragi as intercrop in mulberry gardens. Biofertilizers are eco-friendly, economical and easy to adopt. Azotobacter can reduce 50% of recommended dose of nitrogen fertilizer, whereas WAM can reduce 15 to 20% of recommended dose of phosphatic fertilizer without any adverse effect on leaf yield and quality. Thank you.